Happy end of fall semester and welcome to my local expert presentation about RNAi. So what is RNAi? This technique is a mechanism of post-transcriptional gene silencing. It utilizes double-stranded RNA to target specific mRNAs for degradation, thereby silencing the expression of these undesired proteins. RNAi is naturally found in plants, animals, and fungi, and this discovery brought new knowledge of how cells guard their genomes, which led scientists to develop new strategies for blocking gene function. It has also given rise to various RNA-based strategies for therapeutics in medicine. So how does RNAi work? RNAi is a two-step process, the first step of which has two different possible pathways. In this first step, dsRNA is cleaved to produce small interfering RNAs, or siRNAs. And in step 2, these siRNAs are utilized to target corresponding mRNA for destruction. The two possible pathways to create siRNAs are, first, by delivering synthetic effector siRNAs via various carriers, such as by utilizing attenuated viral vectors, to target cells as preformed 21 base duplexes. This method is simple and effective, but it usually does not last very long. Alternatively, DNA can be used to create short hairpin RNAs, or shRNAs, which are then exported from the nucleus to the cytoplasm and processed into siRNA. This gene therapy introduction is very stable and allows for a much more long-lasting effect from one treatment with viral vector-delivered genes. Processing of this dsRNA into smaller fragments is initiated by the dicer enzyme. Once step 1 has been completed, the siRNAs are incorporated into a complex known as the RNA-induced silencing complex, or RISC, which utilizes a multi-component nuclease. RISC takes the unwound siRNA and the nuclease and guides them to the target mRNA for destruction. So what are the uses and advantages of RNAi? The greatest advantage of this technique is that RNAi is highly specific. Only genes sharing the exact sequence with the dsRNA are affected and inhibited. There are also many uses for this technique. It can systemically turn specific genes off to see what happens in an effort to learn the function of said genes. And scientists have discovered that this plays a large role in the immune system of plants in protection against pathogens as well as in self-gene regulation, which both can be further optimized for use in agriculture. As a matter of fact, in plants, viral infection often activates and upregulates expression of multiple RNAi-related genes. RNAi can also be used for a wide variety of therapeutics in human medicine, including treatments for cancer, Alzheimer's, Parkinson's, and age-related macular degeneration. It can also be utilized to create antiviral drugs against HIV, hepatitis B and C, and RSV. In Badri et al. 2021, researchers utilized RNAi technology to create a therapeutic drug designed to inhibit production of the C5 protein, which plays a major role in complement-mediated diseases. As you can see here in Figure 6, the levels of C5 found in the blood over time decreased more and more with each higher dose of the drug. The closest comparison to RNAi technology is CRISPR. In fact, while they have different mechanisms of action, they are incredibly comparable to each other in end result. To evaluate which method was more efficient at systemic knockouts to determine function of gene expression, authors in Morgans et al. 2016 performed parallel RNAi and CRISPR screens on various genes that control growth in human chronic myelogenous leukemia cell line K562. Lentivirus vectors were utilized to introduce the single guide RNA, or sgRNA, and shRNA into the cells. They found that individually, these two techniques performed with roughly the same amount of accuracy. However, the errors that each technique made were predominantly unique from each other. This makes sense given that while both methods are utilized for similar outcomes, the pathway they each take to get there differs. So it is logical that they would have some errors in common and many that are different. Additionally, researchers found that when they analyzed the data from the two techniques together, errors and all, combined they had a much better result than they did separately. This shows that while CRISPR is a comparable technique to RNAi, there's unlikely to be a situation where one is ever more perfect than the other to utilize. More recently, another group of researchers has taken this combination a step further. Huang et al. 2020 utilized RNAi to control and enhance the function of CRISPR, and the effects of RNAi were further enhanced by utilizing the small molecule N-oxygen. Instead of utilizing siRNAs and RNAi, though, this team utilized artificial microRNAs, or miRNAs. As you can see in Figure 7, they found that these miRNAs can be utilized to inhibit the activity of CRISPR entirely, as well as inhibiting the activity of specific sgRNAs. Because of this, miRNAs are capable of reducing the off-target effects of CRISPR, and they can also increase the targeting efficiency of CRISPR by limiting miRNAs that are naturally found in the cell and can interfere with CRISPR. So it sounds great, but is it perfect? Sadly, no. While these inducible RNAi systems offer a clear advantage over constitutive knockdown technologies, each requires multiple rounds of selection for separate vector components to generate stable cell lines. Also, as previously mentioned, viral infection often activates and upregulates expression of multiple RNAi-related genes in plants. But the initial natural and not induced cue is still largely unknown. Moreover, it also remains unclear how RNAi genes are transcriptionally regulated, although post-transcriptional control for some RNAi genes has been described. It is also not perfectly accurate, similarly to CRISPR, so there are risks there as well. Overall, though, RNAi is an incredibly useful tool in the lab and in the clinic. I really hope you enjoyed learning about it today.